This is everything you need to know for IB Bio, section B1.1, Carbs and Lipids. Just like in geometry, Co is going to be your best friend. Carbs and lipids are both made up of carbon, oxygen, and hydrogen. You can remember that through COA or COH, but they have different properties because of the form of the molecule. So carbs and lipids are widely known as like energy stores, like with carbohydrates with starch and glycogen, and lipids with like fats and oils. Both of them are like very good for energy storage. They're both chemically stable and they release energy when they're oxidized by cellular respiration. So if you don't know already, carbon is one of the most essential elements to sustain life. Without carbon, there would not be any life. But with that, basically the function of carbon is basically limitless. So carbon can form covalent bonds with other atoms, which is sharing electrons, where negative is attracted to positive. So covalent bonds are one of the strongest types of bonds, and carbon atoms can have either 4 single covalent bonds, 2 single covalent bonds and a double, or 2 double covalent bonds, but those are usually found in unsaturated fatty acids. And they don't just have to form these covalent bonds with carbon, they can form this with like other elements such as like hydrogen, nitrogen, phosphorus. If they bond with 4 atoms in another element, I'll put an example right here. This is methane, which the chemical formula is CH4. See how carbon is in the middle and then 4 hydrogens are added to that, and that makes methane. Carbon atoms can be linked up by covalent bonds to form like this big long chain, and we see this in fatty acids, and fatty acids can have like up to like 20 carbon atoms. We see this widely in linoleic acid, which I'll put an example right here. It has a really long chain of carbon atoms formed by covalent bonds, but it'll never be flat like this because it's always trying, the covalent bonds formed by the carbon atoms are trying to spread out as far as possible, so it can form a tetrahedral shape, and so it won't actually be flat, it'll more be like a zigzag, which is like this, looks like this. Okay, now onto macromolecules. They're composed of like a lot of atoms and have a really high molecular weight. The main classes that we like always focus on in living organisms are polysaccharides, polypeptides, and nucleic acids. Polysaccharides are just like many sugars, and each of these are made by subunits linking together into a chain. Subunits are monomers and chain is polymer. To remember that, mono means one and poly means many. But the chemical process that links a monomer to a polymer chain is a condensation reaction. This reaction is when 2 molecules are linked together and that releases like a smaller molecule. With polysaccharides, nucleic acids, and polypeptides, the smaller molecule is always water. And the water is produced from the removal of the hydroxyl group from one of the molecules being linked and a hydrogen from the other. So to construct polysaccharides, nucleic acids, and polypeptides, you're going to need energy. This is usually from ATP, which that is a whole another thing we can get into. So a disaccharide is when 2 monosaccharides are linked together, and a polysaccharide is when multiple monosaccharides are linked together. An example of a monosaccharide is like glucose. If you see anything with the ose on the end, that just indicates that it's sugar. So glucose is used to make like glycogen, starch, and like cellulose. Something that you have to remember is that glucose is linked up with glycosidic bonds, and that the COC linkages are formed by condensation using hydroxyl groups. This is a glycosidic bond in 2 glucose molecules. And if we went through dehydration, that's where we would get water from. Monomers are like the building blocks of life, so they're not just going to be used once when made into polymers. Polymers can be deconstructed using hydrolysis into monomers to make new polymers or used for energy. So hydrolysis reactions can deconstruct polysaccharides, polypeptides, and nucleic acids into monosaccharides, amino acids, and nucleotides, which amino acids and nucleotides is also like a whole nother thing that we can get into. And this all happens in digestion, which obviously in animals just like us, like it happens in our gut, like our stomach, where we digest like our food and stuff. So monosaccharides. Mono, 1, saccharide, sugar. They have about like 3 to 7 carbons, and the most famous monosaccharide is glucose. Some of the properties of glucose is that it's soluble in water, which obviously means that like it dissolves in water, as well as if it's soluble in water, it's also soluble in blood. With glucose being so small and being easily transported, it is easily circulated throughout your blood and dissolves in the plasma. As well as it's stable and it yields energy when oxidized, so it makes like a good substrate for respiration. Glycoproteins. So glycoproteins are formed with a polypeptide with a carbohydrate attached. In most cases, the carbohydrate is an oligosaccharide. Glycoproteins are a component of plasma membrane and are used in cell-to-cell recognition. An example of this is ABO antigens in red blood cells. So there are ABO glycoproteins in red blood cells. There's no like we don't know like a function for it yet, but we do know that it affects blood transfusion. And the oligosaccharides are called O, A, and B. And everybody has like one or two of these oligosaccharides. So say like a oligosaccharide is transferred into a person and they don't already produce that, the blood is rejected. So that's like how that works. However, O oligosaccharides don't cause rejection problems. It's only A and B that do that. Now we're going to be talking more about lipids. Lipids are widely known as hydrophobic because obviously like our butter and like our oil, like if you try to mix it in water, it's not going to mix in because it's hydrophobic. But it's actually like a pretty misleading term when it comes to lipids, because lipids, it's not like they're repelled from water, they're just more attracted to non-polar solvents, which like an example of that is like acetone or stuff like that. Some examples of lipids are fats, oils, waxes, and steroids. Do not forget steroids. Formation of triglycerides and phospholipids. So this is a triglyceride. It's one glycerol with 3 fatty acids, and that makes a triglyceride. And this is linked using a condensation reaction, and 3 water molecules are produced. And don't forget that the linkage between the glycerol and the fatty acid is called an ester bond, almost like Easter, but ester. Phospholipids are similar to triglycerides, but instead of 3 fatty acids, they have 2, and in that spot, they have a phosphate group. And the phosphate group is hydrophilic, so phospholipids are partly hydrophilic, partly hydrophobic. An example of this is like cocoa powder, because if you try to mix it in, like at the start, it doesn't really mix in, but the more you mix in, it will eventually mix in. Keep up with me, we're almost done. Fatty acids have an unbranched chain of carbon atoms as well as hydrogens bonded covalently, so then it makes hydrocarbon chains. The acid part of the molecule is the carboxyl group, which is at one end of the chain, and then the methyl group, which is at the other end. If these fatty acids have all single bonds, then it's saturated. If the fatty acid has one or more double bonds, then it's unsaturated. If the fatty acid has one double bond, then it's monosaturated. If it has more than one double bond, then it's polyunsaturated. Lastly, steroids. So steroids are a group of lipids with molecules that are similar to sterol. They have 4 fused rings of carbon. They have 3 cyclohexane rings and 1 cycloheptane rings. And they have 17 carbon atoms. They're mostly hydrocarbon and they're hydrophobic. With them being hydrophobic, they can easily pass through the phospholipid bilayer, which helps them enter and leave the cell. An example of a steroid is like testosterone. I hope this all really helped. Go pass your test.